Could cloning make immortality possible? It sounds like science fiction, but scientists actually tried it, and they kept at it for 20 years. 58 generations of cloned mice, more than 1,200 individuals that were nearly genetically identical, over 30,000 cloning attempts. And the result? By the 58th generation, every cloned mouse died on the second day after birth. Not a single one made it past day 2. This study was published on March 24, 2026. In 1996, Dolly the sheep shocked the world. Not long after, the world's first cloned mouse, Cumulina, was born, and the scientists behind today's story were part of that history. This was the team led by Teruhiko Wakayama at the University of Yamanashi in Japan, the same group involved in creating Cumulina. Cloning one mouse is no longer that surprising, but this team asked a far more radical question. What happens if you clone a cloned mouse, then clone that clone, then clone the next one, again and again, generation after generation? In theory, as long as each generation survives long enough to provide another somatic cell, the original genetic information could keep being passed on. Wouldn't that amount to a kind of genetic immortality? In 2005, they decided to find out. They started with a single female mouse, then created the first cloned generation, the second, the third, and kept going. For the first 25 generations, things did not seem to be getting worse. If anything, they seemed to be improving. By the 26th generation, the cloning success rate had climbed to 15.5%, the highest point in the entire experiment. These cloned mice also appeared mostly normal, and their lifespans were roughly comparable to those of ordinary mice. Early genome analyses did not reveal any major problems between the first and the 25th generations. Around 2013, based on interim results, the team even suggested that with further technical improvements, repeated cloning might be sustained for a very long time. For a moment, it must have seemed as if they had touched the edge of immortality. But after the 27th generation, things began to change. The cloning success rate started to decline steadily. At first, it looked like a slow leak. Later, it became a collapse. By the time they got past the 57th round, the average success rate had fallen to just 0.6%. And then came the most dramatic result of all. Every 58th generation cloned mouse died on the second day after birth. The entire lineage came to an end. So what went wrong? The answer was hidden in the DNA. When the researchers performed whole genome sequencing, they found that new mutations were accumulating in these repeatedly cloned mice at about 3 times the rate seen in mice produced through normal sexual reproduction. And these were not just tiny scattered errors. The most dangerous changes were larger structural variants, major deletions, rearrangements, and in some cloned lines, even the loss of an entire X chromosome. You can think of it like making photocopies. The first copy looks a lot like the original. The second still looks fine. The third may still be usable, but if you keep making the next copy from the previous copy over and over again, the blur, the noise, and the defects begin to build up. Eventually, the image degrades so badly that it can no longer function as a faithful copy. That is essentially what happens when mutations keep accumulating through repeated cloning. The researchers then carried out another crucial test. What if these later generation clones were no longer used for cloning and instead were allowed to reproduce normally through sexual reproduction? The answer was striking. These later generation cloned mice did show a clear decline in fertility, but once the lineage returned to sexual reproduction, things began to recover. Their offspring had placentas that looked much closer to normal, and by the grand offspring generation, fertility had improved substantially. In other words, sexual reproduction appeared to dilute or reset some of the damage that had built up through repeated cloning. This study tells us at least 2 important things. First, with current technology, repeated cloning cannot preserve mammalian genetic information indefinitely in a stable way. Second, if the goal is to preserve valuable genes over the long term, the best strategy is not to keep cloning one generation from the next. A much more reliable approach is to cryopreserve somatic cells from the original donor and clone directly from that original source each time. 20 years, 58 generations, more than 1,200 mice, over 30,000 attempts. An experiment that set out to probe immortality ended with death on day 2. And that leaves us with one final question. If one day technology really can prevent this kind of genetic damage, would humans use cloning to chase immortality? If you enjoyed this video, don't forget to like and subscribe. I'll keep bringing you more cutting-edge research in future episodes. See you next time!